Hello everyone, my name is Atif and today I will talk about the ultrasound optical imaging technique. We know that the light is consist of different type of frequencies and respective to that frequencies there are different wavelengths. And if we look at our brain anatomy, then our brain is also consist of different layer and each layer is consists of different type of cells, nuclei and glia. So when we throw the light on our brain, then the response of different layer is different. Some light reflects and some light is absorbed by the cells and some and the somewhere is scattering happens. Actually, the scattering is here the problem because we lose information to the focal point. So in this case, we need to overcome this problem so that we can get the exact informations. And here we can see that what kind of range of light can penetrate in what depth of our brain. So the solution is here, the wavefront shaping systems. Here what happens, we send the guide star inside the target area, and when we put the light, here is a light beam, so then information came out in terms of waves, and it is recorded by the wavefront measurement systems, and then that information is sent to our computing system, and in that computing systems, we choose our desired signals, and then by using spatial light modulator, we send that information back to the targeted area. So the overall, all these informations and the experiment is collected by interferometry techniques. You can see here light is actually coming out from the targeted area, the guide stars, and after that, we just put the desired signals to our targeted area. So for that information, the tapered optical fiber is required to perform that, because tapered optical fiber is actually the advanced form of the wavefront systems. So in that case, the optical fiber contains a lot of different wavelength inside it. We can see here is a different wavelength inside that, and then we, according to our desired area, we hit the regions. For example, if we put this optical fibers on the cerebral cortex, then we know that the cerebral cortex contains different types of layer. For example, if we look at the sensory systems or somatosensory or auditory, then this is not consist of one layers. This consists of 5 to 6 layers, and each layer has its different refractive index and different response to the light. So then according to that layers, we can select our range of our light to according to that layer from which we want to get the informations. And here is another the development that made that the optical fiber contain the light information of many wavelengths. So to make it more specific, so we did the micro and nano fabrications of reflective metals, and we made window to use some specific range of light for some specific layer inside the cerebral cortex. So in that case, we can reach at our exact targeted area with the energy or the wavelength which is required in that area. So in the recent usage of this technique, the neural activity is recorded very successfully in the mouse brain, in the basal ganglia and in stratum regions. Thank you very much.
Hello, everybody. In this part of my research, I'm going to talk about the multimodal approaches which have the electrode array with functional microscopic imaging. So my name is Asiq. Now at this part, let's talk about the electrodes, which are like the conductive electrode that you put on the brain to get the brain signal or the other electrocardiogram data. So some of the research, they use this graphene-based system to develop the opaque version of this micromodal array that can use these opaque properties of their graphene array to pass the light through. So you can use in the optogenetics or other kind of imaging, and on the same part on the real time, you can collect the EEG data. So some of the research has, as you can see on the reference, have used this kind of data to the system to combine both of them together. Now, on the next part of the problem is that the graphene is not as longevity of the graphene have not been researched yet, and there is a problem with the impedance of this kind of multinodal arrays. Now, on the other part, there are, in the future work, maybe they will focus on this part of the research when they can use this multinodal array data set to approach how to process this data, because the data set is long and big. On the next part, the optical imaging and functional magnetic resonance. So there are the two separate things they have used together. So we can use this fMRI to put the part of the image, and then we can focus on the 2-photon or other calcium imaging to exactly focus on which part of the brain we want to map. So combining these two methods will generate a very big more conventional, more better than the conventional systems. So these bold fMRI and optical imaging performed continuously or serially together can render a more better imaging technique. So that's all for my presentation. Thank you for listening.